In class, we learned that the microbial world can be divided into non-living infectious agents, which includes viruses and prions, and living organisms, and that those living organisms are then further subdivided into 3 domains. In general biology, you spent a lot of time talking about these guys, eukaryotes, and you learned a lot about eukaryotic genetics and eukaryotic metabolism. And so far this quarter, we've spent a lot of time talking about the domain bacteria. What I want to do right now is take a look at the domain Archaea and consider what it is about these organisms that makes them so unique and so interesting to learn about. There are organisms in the domain Archaea that can live in places in the world that you might imagine no living cell could possibly endure. And these images show you some of those places. Archaea have been found in geysers, they've been found in lava lakes, and they've been isolated and are continuously isolated from deep sea hydrothermal vents. When you consider that a hydrothermal vent can reach temperatures of 400 degrees Celsius, it's pretty staggering to think that any living cell could withstand that type of heat and that type of pressure. But Archaea are up to the task. So what is it about archaeal cells? What structural features do archaeal cells have that allow them to survive in these really extreme temperatures? If you were to look at an archaeum under the microscope, you would see that it actually looks a lot like a bacterial cell. And in fact, Archaea can have many of the shapes that bacterial cells have. In addition, they also have DNA like bacteria do. They have proteins, and they have membranes that are comprised of fatty acid tails. So what is it about each of those macromolecules? What is it about their protein, their DNA, and their membranes that allows them to survive in these really high temperatures? It turns out that Archaea modify these molecules in such a way that makes them very heat resistant. So now let's take a look at each of these and start with protein. You'll recall from general biology that proteins are comprised of amino acids. And those amino acids come together and bind in such a way that folds this molecule. And it's the folding of the molecule that gives it its overall shape. Proteins are very dimensional molecules, and it's the shape of the protein that lends its functionality. This is a concept that you're going to want to store away in your brain, because anytime we talk about proteins, you have to consider what the 3-dimensional shape of a protein is, because that is what makes that protein functional. Now, the way that a protein is folded is largely due to the types of amino acids it's comprised of. There are 21 amino acids that are the building blocks of protein. And these amino acids can be divided into different categories based on what type of side chain they have. Sometimes you may have seen that side chains are referred to as R groups. It's the same concept. Some amino acids have electrically charged side chains, so they can be positively or negatively charged. Some have uncharged side chains, and some, like these down here, have hydrophobic side chains. If you were to consider the generalized schematic of what an archaeal protein looks like, it would be something like this. So imagine here that these are all amino acids that are strung together. AA represents amino acid. And these lollipop type structures that are sticking up from the surface, these represent the side chains. So you can see that these are organized in a very special way. The partially negative side chains are on the outside. There's a positive side chain here, a negative side chain here, and a bunch of uncharged and hydrophobic side chains here in the middle. If we were to consider how such a sequence might fold, it would look something like this. So you can see that the positive and negative charges were attracted to one another, and when they bind to one another, they form a structure that's known as an ionic interbridge. And this helps stabilize the overall folding of the protein. And in coming together, it also shields the hydrophobic residues from the aqueous environment. Now, if this protein were to come unfolded, let's imagine for a moment that the bonds between the side chains got broken and the protein started to unfold or denature. What would happen is that these hydrophobic residues that are located in the middle here, hydrophobic residues are water-fearing, and so suddenly thrust into the presence of an aqueous environment, they would repel water. In repelling the water, they would encourage this refolding. And that refolding would then further be bolstered by this ionic interbridge, which I like to think of as kind of a seatbelt. So it clicks it together. So the bottom line here is that archaeal proteins are able to withstand heat because they have core amino acids that are highly hydrophobic. And another way you can think about this is to consider that water carries heat. So if proteins are able to shield themselves from water, then in effect they're able to shield themselves from the heat. And so Archaea have proteins that are pretty exceptional. The next structural feature we're going to consider is archaeal DNA. And there are three basic mechanisms we're going to look at that archaeal cells use to preserve their DNA in extreme temperatures. Here's your generalized schematic of DNA. So just to reorient you here, you have your phosphate sugar backbone and your nitrogenous bases that form the rungs of the DNA ladder. You'll remember from general biology that adenine pairs with thymine, and it does so with 2 hydrogen bonds, and that guanine pairs with cytosine with 3 hydrogen bonds. It turns out that in Archaea, they have genomes that are really rich in C's and G's. And because they're rich in C's and G's, they have more pairings that have an additional hydrogen bond. And that allows the DNA to be more stabilized. So that's the first mechanism, is that archaeal DNA is largely GC rich. The second mechanism we want to consider is that the negatively charged backbone gets stabilized by the presence of cations. So you'll recall that the phosphate molecule carries a negative charge, and it's this negative charge that confers an overall negative charge to DNA. In archaeal organisms, there are molecules present, such as this one, potassium cyclic 2,3-phosphoglycerate. It's not as important to me that you remember the name of this molecule as it is that you realize that potassium is an example of one of these cations. So when the positively charged potassium binds to the negatively charged DNA, it neutralizes this charge. And because DNA then has a neutralized charge, it's less likely to interact with other molecules around it. And that helps stabilize the DNA. So we have a genome that's rich in that's high in GC, and we have a sugar phosphate backbone that's stabilized and neutralized and charged by positively charged cations. Now the third thing that makes the DNA particularly heat stable is the way that it's coiled. So the DNA gets gets twisted and twisted and coiled and wrapped around these proteins that kind of look like tennis balls here. Gets wrapped around these proteins and then further supercoiled so that it's extremely compacted. This might look familiar. Eukaryotic organisms do this with proteins called histones. So archaeal organisms have a similar type mechanism. Their proteins are histone-like. So they have proteins that bind to the DNA and coil it up so that it's extremely compact. Now, in addition to heat-stable proteins and heat-stable DNA, archaeal organisms also have heat-stable membranes. So we're going to take a look at how these membranes are organized. But before we do that, let's revisit what your typical bacterial or eukaryotic membrane looks like. You'll remember that in bacteria and eukarya, there's a phospholipid bilayer. So you have your phosphate heads, which are hydrophilic here, and your lipid tails that come together and are held together by hydrophobic interactions. If a bacterial or eukaryotic membrane was exposed to really extreme conditions, these molecules would start moving very quickly. The type of bond that holds these tails together is called a hydrophobic interaction. And hydrophobic interactions are relatively weak. So when heat is applied to this type of bond, they break pretty easily. And the membrane peels apart. Further exposed to heat, these would continue to break apart. So this is what would happen to us if we dipped our toes in a lava lake. Our membranes would basically disintegrate. And we would be very, very sad. Now let's compare this phospholipid to the type of fatty acid chain that you'll see in an archaeal cell. And let's look at it in context of its membrane. So what you can pick up right away from comparing a bacterial or eukaryotic membrane to an archaeal membrane is that in Archaea, their heads are covalently linked to one another. So they're linked all the way across, and they're linked by covalent bonds. Covalent bonds are the strongest type of bond. Hydrophobic interactions are pretty weak. So, the archaeal membrane is made of lipids that have covalently linked heads. Bacterial and eukaryotic membranes, on the other hand, are comprised of two layers of phospholipids that are not covalently linked. Now we want to take a closer look at the types of fatty acid tails that these two different membranes have. Bacteria and eukaryotic membranes, if you were to look at their structure, look something like this. So you can see they consist largely of single bonded carbon-to-carbon chains. If you look more closely at these bonds, however, you'll see something like this. You'll see the presence of a lot of double bonds, and you'll also see a lot of side chains. So it's these hydrophobic side chains with double bonds and branches that makes these archaeal membranes extremely stable. If we were to look at these side chains another way, you would see that they kind of look something like this. So here's this molecule that you saw here. It's the same structure. And this molecule is called isoprene. So archaeal fatty acid tails are comprised of isoprene chains. These isoprene chains are bolstered by these double bonds and all of these side chains. And that makes the membrane of an archaeal organism extremely stable. Another view of this is here, and I don't expect you, again, to memorize the structure of this. Instead, what I want you to do is kind of get the eagle's eye view of it. You can tell from comparing bacterial membranes here on the right-hand side that these are linear, predominantly single bonded structures. And on the left-hand side here, you can see that they're connected from head to head, completely covalently linked from head to head, and there's a lot of branching and double bonding that's introducing all of these kinks. And that makes archaeal membranes extremely resilient in the face of extreme heat.